That works, okay. All right. Hello. So I'm going to talk about independent component analysis of EEG and related data. And it was about 20 years ago that I discovered the value of it. And well, here I am 20 years later. And so let's see now. Where's my advancer? This white thing? Now it says Mac, but what do I do? Which button do I push? This one, the arrow button? Yeah. Okay. All right, so let's start with some basic electrophysiology. And the fact that makes the study of electrophysiology important for brain research is, of course, that electric fields are involved in biochemical processes used by the brain at every spatial scale. But the important thing to remember is that so brain dynamics are inherently multi-scale in a way that none of our technology is yet. And but the thing to remember is that at each spatial recording scale, the recorded signal you get is produced by active partial coherence of distributed activities at the next smaller spatial scale. So for instance, if you recorded from the bouton of a neuron, you would record the synchrony in time and space of molecular events. If you record from inside the cell, you're recording synchronies, the results of synchronies in time and space across synaptic and non-synaptic impinging events. And if you record extracellularly, you're recording the synchrony in time and space of local events, local extraneuronal events and neuronal events. And so on, so forth. If you use larger and larger electrodes farther and farther away from the cortex, you're only recording synchrony. So the term synchronizing, meaning amplitude increase, was used with good purpose by the original neurobiologist to mean that when you see an increase in amplitude here, you're seeing really an increase in synchrony at these smaller scales. Now, the reductionist sort of thinking of science would say that, well, if we understand everything at this level or maybe that level over there in the quantum quarks, then we understand everything. Of course, that is proved impossible. Nonlinear dynamics has shown that the macro events cannot be predicted from any known measurement or any possible measurement of the small events. But recently in the brain, it's been shown that local field dynamics, that is to say at larger spatial scale, also influence spike rate, timing, and particularly spike synchrony in a local area, which in turn must influence the common targets of those neurons, so that there's a mixing of scales, so that field activity actually affects spikes, even at EEG strength. That's been shown now in several experimental studies, but it's been very slow to be picked up by neurobiologists, even theoretical computational neuroscientists, because it goes so much against the long concentration on the spike of the single neuron as being the unit of operation in the brain. Okay, so now here is a movie with 1 million sources. And at some time in that movie, during this period here, some of those sources become synchronous. So at no time does any of the amplitudes of any of these million sources, each mini column in the cortex is modeled as an independent source. At no time does the amplitudes change. There's no change in amplitudes. But look at the change in the scalp potentials when this synchrony, the activities here don't become larger, but they become synchronous. So then this dominates the EEG completely. Whereas here, any interpretation you might have of any kind of fields is all, in a sense, for nothing because it's just a random cancellation and addition of a million different sources. Okay? So again, synchrony is what's going to dominate the EEG, and local synchrony, because local synchrony is supported by local connectivity. So these are the effective sources of EEG, I posit. And Walter Freeman, 40, 50 years ago, began to see that when he put very small arrays, grids on the cortex of animals, and he would see the random field, phase of the random field replaced at certain times by what he likened to pond ripples, and he called phase cones, in which there was a smooth phase structure, an event-related synchronization at the small scale. Recently, Beggs and Plentz have come to this from nonlinear dynamics. They model these as circular avalanche events. And modelers of cortex, with their complex models of cortex that have many free parameters, find that many of the parameter settings has cortex making these same patterns. But now, this is a picture, of course, of the sun taken on this little telescope, by the way, and it's a snapshot of a movie of sun processes. Now you see here, see features at every spatial scale. Here's a large sunspot, here's a different spatial scale, and on down. What is the similar picture if we were to image electrical activity on the cortex? Well, no one knows, really, because the spatial temporal field dynamics of cortex have not yet been imaged on a large enough spatial scale. It will be done probably in the next decade. But as of now, how many small spots, how many large spots, how much small pools of synchrony, how many large pools of synchrony, when and where on the cortex, is really only known indirectly. And the size of these synchronous pools needed to support strong EEG sources really hasn't been imaged at all. Now, of course, these sources, each of which, by the way, has a projection, a very broad projection to the scalp that's equivalent to that of a tiny little battery, a tiny battery, infinitesimal battery, in fact, called the equivalent current dipole. But again, the fields here sum at the scalp electrodes. So each scalp electrode sums the activities of many brain sources and also non-brain source processes like eye movements and so on. So this is the EEG fallacy, that somehow the EEG streams up from the brain directly through each channel. It's so easy to fall into that, and I've seen decades of researchers falling into that, whether or not they would intellectually agree. They treat the data as if this channel must be something about the parietal lobe, and this has to do, et cetera. Well, what's the actual truth? This is biophysics now. So this is from Zeynep Kalinachar, my colleague, who has the NFT toolbox to make these electrical head models from an MR image. So this is a subject's electrical head model, and I asked her to simulate just one of these effective sources, synchronous across a patch. So this is the source patch, and this is its projection to the scalp. You see it's either positive or negative across the entire head, except for a tiny equator line in which it does of electrodes, which wouldn't receive potential from that source. So this very broad point spread function, it doesn't matter how big the source is, by the way. If the source is just a tiny equivalent, a single mini column, as we imaged before, or even an infinitesimal dipole, the projection would be the same. It would be still as broad. So there's a very, very broad point spread function by which EEG channels on the head mix EEG processes. Okay, so now this is a quiz for those who haven't seen this before. Okay, look at this. This is, again, a projection of a lot of source activity. So who imagines they understand what the source activity is? Okay, try to think what sources are producing that. How many people are thinking of traveling waves on the cortex? No one. This is a very bright audience. How many people think there must be frontal sources and then parietal sources and occipital sources? No one. Well, you know everything already, so. Okay, this is actually what's happening. So these are two spatially static sources. One is, simulated at 9 hertz. Of course, it's going 20 times too slow. And the other at 10 hertz. And they add, and therefore they cancel. And that produces this apparent. But this is all epiphenomena. So all this that you see here and try to interpret with your eyes is all an epiphenomenon. What happens at the follicular level doesn't really affect the brain. It doesn't affect cognition. It doesn't even help you grow your hair, I don't think. So this is a powerful, I think, example of the EEG fallacy, that if you try to interpret what you read at a single electrode, you're prone to vast mistakes. And Christoph showed that too. Now here is 30 sources, all with EEG, simulated with EEG. And this looks a lot like, this is indistinguishable from actual EEG recordings, except, of course, that it's many times slower than the EEG recordings. So an EEG recording, think of that going 10 times as fast. Now, each of these, again, is each blue or red here, positive, negative, or positive, is again the sum of source activities occurring all over cortex. And just because you see a red patch somewhere doesn't mean that the same sources are producing that red patch in each instance. So in a sense, the scalp map, I say, anything you see in the scalp map is strongly at risk of being an epiphenomenon. Okay, so let's go down to source imaging. So if you have an MR image of the subject's head and you segment the different tissue types with different conductivities, the skull, the brain, CSF, and so on, then you can build an electrical head model that says how currents flow from the source space in the head, in the brain, in particular in the cortex, out to the scalp. Okay, this is a boundary element model. The finite element model is more flexible and so on. Now, given some EEG data or some MEG data with appropriate signal processing and sensor localization in terms of this model, you can come up with an inverse problem. The inverse problem is, at best, a simple map. If it is a simple map representing the projection of a single source area, then in almost all cases, the problem is actually solvable. In principle, the problem is solvable. You can find the patch. But the question is how to get a simple map that represents the projection of a single source. Well, here's the paradigm for independent component analysis, the cocktail party problem. You have multiple noise sources, sound sources at the cocktail party, and you have multiple microphones, each one recording cocktail party noise. Okay, sounds like that. So about 1990, some French applied mathematicians reasoned that these are really, even though you have these mixtures, there's only 2 sources of information now in these channels. So if you just drive the linear unmixing process toward greater and greater independence, more and more distinct information in a technical sense in each of these signals, you can only come back and recover the original signals without knowing anything about the acoustics of a man's voice or the saxophone or having any idea what the sources are. So I read the paper by Tony Bell and Terry Sinofsky 20 years ago almost today, and I had a dim idea that this might work for EEG. So you have sources here, broadcasting, summing at the electrodes linearly. The propagation is near instantaneous, which is easier than in the acoustic case. So perhaps we could recover the time courses of the individual sources, unmixed. And so a week later, we made a bold claim in a paper we sent to NIPS It's said that the ICA can find these distinct effective source activities, and they're simple scalp maps. So the assumptions of ICA are the sources are spatially stationary, at least at the sort of the centimeter scale, and that they're independent. Their time courses are independent. Now, why is that a useful assumption? Okay, here, let's take these to be these patches of synchronous activity in cortex. Now, the excitatory and inhibitory cell networks within these patches are the principal cause of sustaining oscillations and the buildup of potential patterns that are spatially synchronous. This alone, the lack of long-range connections of the inhibitory cells by itself means that synchronous activity, if it starts to spread, should spread across a compact patch, just as Walter Freeman observed. And Plentz can model. The primarily radial thalamic cortical columnar structure also supports oscillations and has very little crosstalk at the thalamus. The long-distance cortical connections in cortex are vastly sparse compared to nearest neighbor local connections. So most connections in the brain, almost all connections in the brain are local. This means that if synchrony begins to spread in another patch, there is very little to synchronize those two. And the idea of independent or nearly independent domains of synchrony is physiologically highly plausible. Now, of course, as I said, each of the projection of such a patch, such a synchronous patch, would be that of an equivalent current dipole, a mathematical small battery located in or very near the patch. Now, of course, some cortical regions are connected, say corpus callosum. And you can imagine a synchronous or a resonance activity occurring synchronously in both patches if they're bidirectionally coupled. In that case, of course, you could think of this as one information source in the data, and its projection to the scalp would be that of two current dipoles located at either end of a white matter tract, for instance, corpus callosum. Okay, so what do you see when you actually run ICA? We ran ICA on 100 channels of data, a half hour of 100 channel data during a 2-back task. Here are 9 of those channels shown, and here's 15 seconds shown. And here are 9, I believe, independent components of that data shown for their activities in the same 15 seconds are shown. So what we see here when we mean temporally distinct, that means from knowing the value at time t of any of these, you cannot predict what the value is at time t in any of the other components. They're all independent. They give you there's no information here about what the value is at any one moment here. They're distinct, and they're functionally distinct as well. So, for example, here is ECG, or EKG if you prefer. That is actually in almost all the EEG channels to some extent. Although the spike here can be seen in this case in some of the channels, but certainly not in others. So only ICA could pull this out. You can't find it with frequency domain filtering or any other kind of filtering. Here is an EMG source. And when we say component, we mean a component process, a source, an effective source. And this comes from a postauricular muscle that has muscle activity and then spiking. Here is at the same time a subspace. ICA separates the data into independent subspaces. Obviously, these aren't quite independent. This spike occurs all before these. These are eye blinks. And so this is an independent component. This is a dependent subspace, but it's independent of everything else. So ICA separates things that have some change in spatial pattern in a reliable way into multiple components. It finds some basis for this dependent subspace, but it makes it independent of everything else. Here is frontal theta, which you now can look at trial by trial with high signal to noise ratio. Here is another frontal theta, independent of that. And here is an interesting bilateral occipital alpha burst that occurred after every trial. The subject here was seeing a letter, making a button press, getting a beep or a buzz, and then resting for a few hundred milliseconds. And there was alpha burst in his lateral occipital cortex. Of course, one of the first things we realized is that you can separate out non-brain processes. Here are eye blink, an eye blink process, or eye blink component, we would call it. Here's a component process that accounts for lateral eye movement activity from saccades, and here are they in the single trials. Here is a polarity reversal, a little too high, I think, in our early co-registration for a postauricular muscle. And you see in the first part of the experiment, the muscle was very active, then it became quiet, and then turned on again and became quiet. I doubt very much the subject would have been aware of that, but that's what we see. And here's the typical EMG spectrum. Now these are all pulled out of the single session. And here is the ECG again. Here is from a 256 whole head array. Here are multiple individual neck muscles with their EMG spectrum. And if you look at Gray's anatomy, you can look at the angle and orientation of this and believe that you see the muscle, that you can identify the muscle from the anatomy. No one has done a dissertation to actually prove this yet, but I think it's pretty clear. You can actually make a movie of the neck muscles during a evoked acute head turning experiment, but this doesn't seem to be working. What we find here in this trial, these neck muscles become stiff, and then we have red balls that appear here to show that. But before the head turns, this is a stiffening in response to the cue. Not these muscles in the back of the neck is what's turning the head. I'm sorry, this doesn't work. Now, I found this funny picture. You know, this is what I would call, if this were an EEG subject, I would call this non-stereotypic EEG noise. So eye blinks always show the same pattern on the scalp. They're concisely captured by a single component, usually by ICA. But, you know, if the subject were doing this, even for a second in your data, hundreds of maps would be spewed into your data that never appeared again. They were independent of everything else, and ICA would be confused. So what you have to do is you have to get rid of non-stereotypic noise from your data. So you have to excise that from your data. Okay, so when under good circumstances, if you have enough data, enough channels, you can not only capture all those non-brain processes, but you can capture brain processes. And here is an example from that same session. This is the scalp map, the scalp projection, and its equivalent dipole, which fits in our head model, in our somewhat crude head model, extraordinarily well. So this is completely compatible with a source in a patch of cortex. Here is a source that is completely compatible with a source in two patches of cortex that are bilaterally symmetric in position, if not orientation, and therefore plausibly could be coupled by white matter tracks or by common input. Here is a frontal medial midline source, which shows lots of theta activity during tasks requiring concentration. Now, I'm sorry, this looks complicated. These 30 maps are the largest independent components obtained in one decomposition of a data set. So let's take this source map and we calculate its projection to the cortex, the percent of variance explained by this source on all the channels. And you see it's maximally projecting to one channel. But at that channel, only 30% of the variance of that channel is explained by that source. Here's another example. Again, this is a map of variance accounted for on the scalp, and there's one channel that receives most of this, but only 30% of its variance is accounted for by this underlying potential source process. Here's another effective source. Again, this map of variance accounted for, and again, 30% is explained. So in all these and typical cases, only at most 30% of scalp map signal variance in any channel in a channel is produced by one brain source. So 70% of the variance is produced by many other sources. So the signal to noise ratio of EEG is less than 0. Now that's not quite true for electrodes placed right on top of muscles, where maybe 60% is accounted for, or electrodes placed very near the eye, where nearly all of its recording is dominated by these lateral saccades in this case. So, by the way, this decomposition, I would look at it and say this is not very good. Why? We see many dipolar components here, but we see all sorts of odd things that cannot be the result of a single source process. So I would tell the student in this case, or the researcher, please take out some of the noisy episodes in your data and do ICA decomposition again, because these cannot faithfully represent the projection of any source. Now this is a little bit of spatial math. Here is the block of data as a matrix, channels by time, and multiplying it by unmixing matrix, that means unmixing filters, learned by ICA. And what ICA does is learn these filters from the data. That multiplication produces the activations, the time courses of the component processes, which are independent. You can also get the projection of each of those processes to the scalp in the form, cartooned here in a form of a scalp map. This matrix, this mixing matrix, is actually the mathematical inverse of the unmixing matrix. So you can derive it directly from the unmixing matrix. And then the product of these activations fed through these scalp maps and then summed at the electrodes produces exactly the scalp data. So this is a linear decomposition, like in form to other linear decompositions such as PCA and so on, many of them, but with its own purpose of making these independent. So, okay, where do the equivalent dipoles for these processes end up? This is from a group of, say, 30 subjects performing a visual working memory task. Well, and then this represents probability of finding a source dipole, an independent component source dipole in every spot in cortex. So we see that probability is high in this group of subjects during this task in lateral cortex. There were distinct signals that ICA focused on in most of the subjects. Here's anterior cingulate. Well, a difficult working memory task, yes. Here is hand somatomotor cortex. Again, they were waiting to press a button in most of the experiment, and under which circumstances the distinctive mu rhythms arrives. Here in some, but not as many of the subjects, we found posterior cingulate or precuneus sources. But now let's change the task, and another group of subjects, of course. There's an auditory novel oddball, so boops and beeps and very odd novel sounds. And what we find is we still get visual sources, probably having to do with alpha activity. But we see this distinctive source in nearly all the subjects that has to do with the response to this novel stimulus. And let's take a third task. Here, the subjects had their eyes closed, and they were asked to imagine various scenarios in which they would experience strong emotions, such as anger, love, hate, fear, jealousy, and so on. Well, their eyes were closed, and very likely there was alpha activity that of course, attracted the attention of ICA, if I want to anthropomorphize it. There's sort of distinct signals arising in the occipital cortex. But in these subjects, and not in the other experiments, in many of the subjects, there were, of course, sources and distinct signals arising in lateral inferior frontal cortex, which, of course, is associated with emotional processing. And again, they were holding a button, waiting to say, okay, I've had enough of this scenario, pushing it, and so producing mu rhythms. And no hotspot in anterior cingulate. So they were imagining all sorts of complicated scenarios, including ones introducing fear, and yet there was there was a wide distribution of source processes in individual subjects, but there was no tendency for a source to appear in anterior cingulate. Very interesting. Okay, so here's an example. This would be a cluster across subjects of left mu processes and right mu processes. This is how we began, by seeing these and say, okay, clustering must be a good idea to do when you're looking at group data. So this is in EEG lab. This is the dipole fitting clustering facility that you can cluster on dipole location and on spectrum, or on ERPs, or on time frequency measures. Here is a variant of that, measure projection analysis, for which a toolbox is available by Nima Bigla-Shamla. And then here is, I believe, the future. This is not using the dipole representation of these sources, but actually solving to the cortex of the individual subject using their MR-derived head model, and then co-registering all the cortices using Bruce Fischl's scheme in FreeSurfer, and then clustering on the topological cortical surface, smoothed out here for visualization. You can obtain very tight clusters in functionally labeled areas. And I think that's the future of high-definition electrophysiology. Now, this is a very important study of a few years ago. Jason Palmer contributed to us a measure of how much mutual information is squeezed out of the data by moving from channels to component processes. And you see almost PCA squeezes out almost all the data, all the mutual information, and it makes everything orthogonal. But a few percent more, all these ICA methods manage to squeeze out a few percent more when applied here to 70-channel data sets, a dozen or so of them. And this is the number of near dipolar, component processes whose scalp maps are nearly resembling an equivalent current dipole, something like this. And you see there's this linear relationship between the effectiveness of ICA technically and actually making things independent, or finding things that are independent, and then recovering these sources that are completely equivalent, and they're completely compatible with a single source patch. Of course, when we did this, we were very lucky to find that the algorithms we started with and the Amica that Jason Palmer derived in our laboratory are really the best for the purpose of, for this purpose. Okay, so here is some asides on applying ICA to other types of data. Here was the slide where we said that scalp signals are not source signals because they represent the sum of all the sources. But what happens when you put down a grid of ECOG electrodes? This is from an actual grant application by a neurologist who is also a physicist. And he explaining that when you put an electrode on the cortex, it's sensitive to the column of tissue directly below the electrode. But what is missing here? Lateral volume conduction. The brain is wet. And yet the entire field of ECOG, both clinical and scientific, has ignored the lateral volume conduction between ECOG electrodes. So here is ICA applied, well, this will do. Here is ICA applied to a unique data set we got from Greg Worrell in Minnesota that had some 30 scalp channels as well as about 100 grid and strip channels on the cortex. We had to make a head model with the hole in the skull, Zeynep did, and she had to put in the plastic layer in which the electrodes were embedded. And then we ran ICA on the joint data, on the EEG and ECG, because it's all linear, it's all linearly related, so ICA will find projections both to the scalp. In this case, we visualize them on the sheet, and again, visualize them on the brain surface. And you see the spread of lateral spread from a source to many electrodes. And this shows a dipolar source that is an effective source that is in the sulcus. So it projects laterally. Again, it projects to the scalp, across the whole scalp as usual. But it's located between the electrodes and even if an electrode was located exactly there, it would receive nothing from this source. So ICA can be used wherever its assumptions are valid. And I think the application of ICA and electrical head modeling to invasively recording data is something that definitely will be standard in the future, but it's done by almost no one currently. Here is simultaneous EEG and MEG data sent to us from Frankfurt. So 300 channels of MEG, 64 channels of EEG, and we used Amica, that powerful ICA, on all 370 dimensions of the data, and it found components that were synchronously EEG components and MEG components. Now for this more or less tangential EEG component, it found a strong MEG counterpart or corollary and you see the right-hand rule from physics here. This is exactly as it should be. And this source actually coded or produced a visual evoked potentials to each of these visual stimuli in the experiment. Now this source was somewhat more radial in the EEG, and the map was also learned for MEG, but a little less well because there wasn't as much MEG associated with this source. And this had to do with the button presses. These are the button presses in the single trials. So this showed an evoked response around the button presses. But now this source was a frontal EEG source that was almost radial. And you see the MEG map is very poorly defined here. Why is that? Because there's almost no power in the MEG whatsoever for a radial source. So when we looked across our brain sources, and this had to do, although it's not shown very well in the ERP, it had a lot of frontal theta in the experiment. So what we did is we took all the brain-like processes we found in this joint data set, and we plotted the ratio of variance explained in the EEG versus variance explained in the MEG against the angle to radial of the EEG dipole. So for sources that were radial, MEG contributed almost nothing. Only a few percent of the power of the variance explained in the EEG was explained by the MEG counterpart, as expected. And for those few sources that were tangential, this is going to get worse and worse, I think, during the day, this thing. The EEG and MEG were exactly equal in strength. That is to say, the same percentage of variance was accounted for by the EEG portion of the component process and by the MEG portion of the process. Again, those signals must be linearly related. ICA here found the scaling property by which you could compare them. And as the physics dictated, it was related to the direction of the EEG dipole, of the source, actually. Now, here is the same idea applied to high-density EMG. So the subject was making movements in various directions. We ran ICA, and here is a muscle. So the muscle components represent resemble a dipole at the insertion of the muscle into the tendon. And so now, with the signal to noise that we get from that, we can look at the subject's EMG from that particular muscle as well as from all the other muscles in the arm, or most of the other muscles in the arm, in the single trials or in the single conditions. Now this is the last thing I'll show. This is something that's not even published yet. But it's very exciting because I think it shows a way forward to high-resolution EEG imaging. The greatest obstacle to high-resolution EEG imaging is not knowing the skull conductivity. The skull being much less conductive than the brain smears out the potentials. You have to know how conductive the skull is, or you cannot accurately localize the EEG. You can localize the angle, typically, but you can't localize the depth, and the error may be up to 3 centimeters, as our simulations showed. So we've developed a method called SCALE, Simultaneous Conductivity and Location Estimation, in which we either starting with one very conservative assumption about brain, about skull conductivity, or a much more generous one, in the sense this is a very hard-headed person, and this is a thin-skulled person, applied to the same data and applied to a number of the independent components, processes, treating them as if they are sources in the brain and using their scalp maps to localize and looking for sources that are, as we expect, compact. In fact, our algorithm is called sparse, compact, and smooth. We adapt the head model until it converges from either of these two widely disparate starting points. It converges, and the positions and the source locations of these independent components are learned along the way. So compare this, to the kind of minimum norm, diffuse, low resolution, all the low resolution cortical imaging methods. This, I believe, will be shown to be high-resolution cortical source imaging. At least we know it's possible. And we look forward to continuing toward the goal of having available, particularly for clinical investigations, diagnosis and research, and treatment monitoring, high-resolution looks at the effective sources of the EEG, in which independent component analysis, I think, will play a continued role. Thank you. Oh, and this is the ecosystem of EEG lab toolboxes that many people in the world are using now, and many other labs are contributing to. So your methods you can easily plug into EEG lab and make them available to the world, basically. Okay, do we have some time for questions or comments? So again, yes? No, go to the mic, please, because this is being recorded.
Thank you for your nice talk. I also use the EEG lab toolbox, but when I use it, usually that when we record the EEG, that the maximum we can only record hundreds channels, for example, 128 channels. But then the source in the brain may be that more than that. So that using the ICA, is it, I have concern that if the source is more than the electrode that we recorded, is it a problem that to find the true source in the brain?
Yes, the answer is yes. As I showed, Every synapse is a source. Every mini column is a source. Everything in the brain is a source, but it's not a distinguishable source by any stretch of the imagination. So what ICA finds are effective sources. They've distinct effective sources in the brain. So yes, if you went invasively and recorded at very high density on the cortex, you'd find more sources. You'd find the effective sources at that scale. There also would be, as I showed with the ECOG, effective sources of whatever you recorded at that spatial scale. And if you went still smaller and smaller, you would record more and more effective sources. So again, we're looking at, when we look at EEG, at the sum of effective sources. And those apparently dominate the EEG signals. A few dozen sources dominate the EEG signals. But yes, one cannot hope to record all the electrical activity that's occurring in the brain from a distance and using a limited number of channels. It's just not possible because they're not coherent, they're not synchronous, they're not effective sources. They're indistinguishable.